Um, let's go again. So I'm Calico, and I will be lecturing on AP Psychology every day for today and the next 4 days, so 5 days, and we'll cover all 5 units, 1 unit a day. Um, each lecture will be around an hour 45 minutes, but obviously some are going to be longer than others. This is probably going to be one of my longer ones. So I'm really sorry. This is the only unit we're not doing a review game because I don't know if we have time, but we will for all the other units. Today we're going to do Unit 0 and Unit 1. Unit 0 is basically like research, ethics, statistics, stuff like that. That's going to help you. Why can nobody hear me? Are you guys serious? Okay. Um, okay, cool. Okay, most of you can hear me. That's good. Um, Unit 0 is basically research, statistics, ethics, and things like that that you need for your FRQs. And then Unit 1 will cover your nervous system, brain anatomy, sleep, and then sensation. There's a lot of content that we have to cover today in a short amount of time. So I'm going to go kind of fast. So please, please, please let me know if you need me to go back a slide or re-explain anything like that. Yeah. Before we start, I have to introduce you guys to Five Hive. Five Hive is a volunteer student-run initiative led by Lance. We have over 70 volunteers and more than 10,000 combined volunteer hours. We create resources to help AP students for all different kinds of AP courses. You can find additional resources such as articles and practice questions or exams on our website at fivehive.org. If you would like to contribute to Five Hive, you can do that by creating resources such as articles and questions, hosting lectures like the one you're attending right now, or promoting Five Hive. Take a look at this document that I'm about to link in chat. Also, yeah, I'm gonna post this on YouTube. We're gonna upload it later at some point. Our applications are reviewed on a rolling basis, typically around once every 2 weeks or so. So even if you don't have the time to join Five Hive right now, you can join it at any point in the future. Oh, Five Hive is not in any way affiliated with College Board or their AP trademark. So I already kind of said this, but we're gonna go through Unit 0, which is like all your FRQ stuff basically, and then we're gonna go through all of Unit 1. Okay, so we need to start off with psychological perspectives. Each of these are different perspectives on how to explain behavior and mental processes in psychology. Let's take the example of a child being scared of monsters under their bed. The biological perspective might say that a child has a genetic predisposition for increased anxiety levels or paranoia. The behavioral perspective might say that maybe they saw a scary movie and that made them learn to associate beds with monsters. The cognitive perspective might say that the child was misinterpreting sounds and shifts in lighting and begin to perceive them as monsters. The evolutionary perspective would say something like having heightened sense for potential threats was something important that our ancestors evolved to survive. The humanistic perspective would say something like the child's emotional needs are not being met, which, if you don't remember, is based off of Maslow's hierarchy of needs. Um, The psychodynamic perspective would say that the child is having internal conflicts that are being manifested at this fear, as this fear. Um, Industrial organizational psychology. That wasn't part of the Unit 01. Yeah, it's not tested, but it's something that you can use in your FRQ. So there's a lot of things that aren't going to be tested on in your um, MCQs, but you can use them as evidence in your FRQs. Um, Maslow's hierarchy is a very popular one that people do. Oh, yeah. Okay, sociocultural perspective would say something like um, maybe the culture emphasizes, like, has stories about monsters, and that the people around them are telling stories about monsters, and that the people around them expect children to be scared of monsters, stuff like that. My computer hates me. Okay, so here are some things that psychologists need to consider when they're looking at a situation in order to make accurate judgments. One is cultural norms, which is basically unwritten rules for any specific culture. We need to keep this in mind because the same situation that would be completely normal somewhere in the world might look disrespectful somewhere else in the world. Another thing we need to look at is expectations based on culture. Individualistic cultures emphasize personal achievement, while collectivist cultures value someone's contribution to a group. So, in an individualistic culture, someone who prioritizes their own personal successes would be celebrated, but maybe in a collectivist culture, some people would view them as selfish. Psychologists also need to keep cognitive biases in mind. Confirmation bias is when we come into something with a pre existing belief and we begin to cherry pick information that aligns with our belief and ignore information that doesn't. Hindsight bias is when, after you learn the outcome, you think that it was predictable from the beginning. We do this a lot when something bad happens to us. We're like, oh, I knew it. Psychologists think that we do this as a coping method and to feel like we're more in control of our lives than we actually are. Another type of cognitive bias you need to know is overconfidence bias, and this basically describes our tendency to overestimate our ability. Okay, let's talk about experiments. First of all, for something to be considered an experiment, it needs to use independent variables, which means that the researchers are able to control the environment and manipulate one thing at a time to look for change. Experiments also require random assignment. Random assignment is when you randomly put your subjects into groups, making the groups roughly equal. This minimizes any pre existing differences between your subjects that might have skewed your results. Three types of variables you need to know are independent variables, dependent variables, and confounding variables. In an experiment, the independent variable is the thing you're changing. The dependent variable is the thing that's changing depending on the change in your independent variable. Your confounding variables are all the other things that could possibly be impacting your dependent variables. Ideally, we get rid of confounding variables by controlling environments, but things can slip through the cracks pretty often. We typically use non-experimental methods when it's impossible for us to control the environment or it's unethical. For example, if we want to study injuries, we can't just give people injuries. Like, that's unethical. There are four non-experimental research methods that you need to know. One is case studies, where researchers follow around an individual or a small group of people for a long period of time. We might use case studies to observe rare accidents or super unusual cases we can't replicate. For example, there's been a handful of case studies on children isolated in the forest for extended periods of time. The biggest flaw of case studies is that we're not able to generalize our findings. In other words, because people are so unique, we can't say that whatever happened was because of what we thought it was. They might have a biological predisposition, or something else was going on at the time. When we conduct studies with large numbers of people, we get rid of that uniqueness, which is why we're able to apply our findings. Correlational studies are studies that look at the association between two variables. They don't manipulate any variables or control the environment. An example of a correlational study would be someone looking at a screen time versus grades. Maybe their data says that higher screen time is strongly correlated with lower grades. But does that mean that being on your phone causes worse grades? No. All research is built on the foundation that correlation does not equal causation. A meta-analysis is basically when you take multiple studies and mesh together their findings to get a new set of data and statistics. So, if I'm looking at studies published in different countries on anxiety levels, my meta-analysis might be able to tell us that some countries had higher anxiety levels than others and might output a new estimated global average of anxiety levels. A naturalistic observation is when researchers don't interfere with the environment or or subjects at all. They just let life go on and they record their observations. So, for example, if we want to study what kind of toy children play with the most, we can look at them through a camera or we'll sit far away from them and just write down how long they play with each toy. Yeah, they are also called observational studies. Um, okay, my question for you guys is what do experiments allow us to do that non experimental methods cannot? I'm seeing manipulating variables generalize, measure impact causation. Okay, what is that? Show a relationship through variables. Have use of a control group. Sometimes we use control groups in experiments, but not always. My computer hates me. Okay, what it allows us to do is establish causation. We cannot establish causation in non experimental methods. We can generalize in non experimental methods, just not with case studies, because case studies is just like one person or a really small group of people, so they're not representative of a population. Manipulation of data, I wouldn't say that's what it allows us to do, like that's just what you do. That's the difference of what you do in an experiment versus um, non experimental research, but that's not the outcome. That's different. You can measure impact in non experimental methods. For example, with correlational studies, we can get a correlational coefficient that will basically tell us how related our two variables are. The problem is we can't infer causation because we might have confounding variables that are at play as well. Okay. Um, we need to talk a little bit more about experiments. So you need a hypothesis, and your hypothesis is basically the claim the researchers are making. So in my hypothetical study, my hypothesis is that owning a pet will make you happier. The operational definition is how we measured and defined our variables. In, in psychology, we're constantly trying to measure subjective things. Like, how can I measure someone's happiness? It's not like I can just like stick a 2 up their head and be like, oh, you're 80% happy, you're 40% happy. This is why we use tools like Likert scales to measure these variables as numbers. A Likert scale is a rating typically from 1 through 5, but I've seen all different kinds of numbers. And it's where a participant will tell you how much they agree with a statement. In my study, I might use a Likert scale to measure someone's happiness by asking them to rate the statement, I have experienced a noticeable increase in energy since getting a pet, since I might think that energy is an important part of happiness, and that's how I'm defining happiness. Um, However, when we're looking at things like complex and individual experiences that we cannot turn into a number, we'll conduct interviews. And with interviews, we can compile key similarity. Guys, kill me now. Okay, when we conduct interviews, we can compile key similarities that are reported by our participants. So, for example, let's say that a lot of my participants reported in interviews that they got better sleep or that they had more social battery after they got a pet. Okay, your control group is the group without the independent variable. We use the control group to give us a baseline statistic so that we can measure the difference between our control group and our experimental group and therefore the effect. Your experimental group is the group with the independent variable. Sometimes you might have multiple experimental groups if you're manipulating severity of a treatment, which we see is really common when we're testing drugs. Um, in my example with pets and happiness, my control group would be the people who don't own pets, and my experimental group would be people who do own pets. Can you elaborate on the format of an operational definition? Do you mean like how you should write it in an FRQ or something? Yeah. Okay, so I think all FRQs prompt you on this, or I don't remember which one it is, but they should all have something that's like, what is the operational definition of blank variable? So for my example, um, I'm going to give you the operational definition of happiness, which is my dependent variable. I'm going to tell you the operational definition of happiness was measured by increase of energy and increase of social battery reported by the Likert scale. So like something like that. Does that help? Okay, experimental biases. One type of experimental bias is experimenter bias, where the researcher's beliefs can shift the outcome of a study. This can be subconscious, like if you're having a naturalistic observational study and they're like reporting the stuff, they might subconsciously want to prove their hypothesis, and so they might shift their recordings to fit their ideas and beliefs more. Social desirety bias is when participants might change their answers in order to seem like they're a better person. Like this example right here, this lovely example. If I survey people and ask them how much they smoke or if they smoke at all, they might say they smoke less than they actually do or that they don't smoke at all to seem like a better person. Um, yeah, it started like 20 minutes ago. Okay, one way that we can control for experimental biases is by using placebo. We can either use a single blind procedure where only the subjects don't know what treatment they're getting, or we can use a double blind procedure where both of the subjects and the researchers don't know what treatment the subjects are getting. Experimenter bias. Okay, experimenter bias is when the researcher's beliefs can shift the results of the study. If you need an example, one is like if I'm conducting an interview, I might shift, when I'm recording the responses to the interview, I might prioritize the responses that fit my beliefs more. How you phrase a question? Yeah, that can also lead to biases. AP stats comes in clutch, guys. Okay, research validity. Falsifiability is the ability of scientific claims to be disproven. So, if you have a hypothesis and you cannot disprove your hypothesis, you cannot scientifically prove it to be correct either. Replication is the ability of a study to be done repeatedly and to get the same results or similar results. This is really interesting because we're currently in the replication crisis of psychology. Right now, an estimated only 50% of published psychology and social science studies can actually hold up to replication. Generalizability is what describes to what population and to what extent you can apply your findings. Ways we can increase our generalizability is by getting a larger sample size, increasing the diversity of our sample, and using random sampling to minimize sampling bias. Ethics. Um, if you're using live subjects like animals or humans, you need to consult with an institutional review board to make sure you're not causing any unintended harm or harmful effects to your subjects. Another thing you also need is informed consent or assent. Assent is different from consent, and it's when someone who cannot legally give consent verbally agrees or like gives consent in a different way. We do this a lot with children, so we'll ask the children if they want to. I just realized I'm literally missing an ethic. That's what I was going to say. Guys, you also need to know the right to withdraw from a study. Sorry about that. That just means that after you get into the study, you can leave at any time. No, guys, this is literally 40 slides long. Like, bro. Okay. Protection from harm just means that if you cause harm to your subjects, it will be very minimal and it will be short term. So, like, you can't traumatize your subjects or anything like that. Confidentiality means that you need to keep your subjects anonymous. You can use deception if you're upholding protection from harm. We use deception in things like placebo studies where you lie to your subjects about what group they're in. But if you do use deception, you must debrief, which is where you call your subjects back in to the office or anywhere really, and you explain everything you did to them, including what you lied about, etc. We do have to know a little bit about statistics. You need to know how to calculate mean, median, mode, range, and you also need to know your normal curve as well as the percentage distribution of your normal curves, of your normal curve. Um, Skewness is basically how far away your distribution curve looks from the normal curve. So if one side of my curve is super high and the other side is super low, that's a curve that has a high skew. The percentile rank is the amount of people at or below a specific point. So if I say the percentile rank for getting a B on an exam is 59%, that means 59% of students got a B or lower. In non-experimental studies, the effect size describes how related two variables are. In experiments, effect size describes how large the difference in results is between the two conditions. That's like the scale. So a small effect size is less than or equal to 0.2, medium 0.3 to 0.7, large is 0.8 and above. That's like a general range. I'm actually kind of confused why the range isn't continuous because it's the one that I literally stole off of College Board's website, but that's okay. Just know like generally what's large and what's small. If something is statistically significant, it's going to have a p-value of less than or equal to 0.05, which says that the probability of you getting your results due to chance alone is less than 5%. So if we have a p-value of less than or equal to 0.05, we can be pretty certain that our results are due to our variables and not chance. Your decimal is in the wrong place, and yes, you do have to know that. I'm fairly certain it will at least show up once in your FRQs. Guys, I'm uneducated in the FRQs, but like, you know. Okay, here's my FRQ for you. Your actual FRQ is going to be longer than this, and you're probably going to have graphs as well. Your questions are also going to be kind of harder, but like practice. I'm going to give you guys like 3 minutes to read. I'll give you guys 2 minutes to read it, and then we'll go over the questions one by one. So don't put your answers in chat yet. Read it, and then we'll go one by one. Okay, let's start off with the first question. What branch of psychology are these students in? Likely. You can always work in something else, but behavioral, mm, biological, cognitive. Oh, you have all your answers. Um, this is biological. We're looking at the effects of drugs on um, people, which is like a neurochemical thing. Most of behavioral psychology is about like classical conditioning, learned associations, stuff like that. PhD. Okay. What's the control group for this experiment? Ow. Placebo. Yeah. It's the group of the women who are receiving the placebo. Okay, so yeah, hopefully you guys write more descriptive answers on your actual test, but like for the sake of this, we're just gonna. Oh, I didn't. Oops. Okay, what kind of study are the students conducting? Correlational. Um, double blind experiment. Experiment. Double blind. Okay, yeah, it's a double blind experiment. We know that it's an experiment because you are manipulating the treatment, which is the medication dose, and then we're also using random assignment. If you see random assignment, it's um, what's it called? It's an experiment. If you see random assignment on an FRQ, it's going to be an experiment. We get double blind because neither the researchers or the subjects knew what group they were in, if they were getting the drug or not. Okay, what is one kind of ethical standard being violated? I did not have enough details, so there's a lot more than what I put that you could argue for. Just give me one. Random assignment equals experiment. Yes. Deception needs to be debriefed. Yes. Confidentiality, deception. Um, I wouldn't say deception, I would say debrief. Yeah. Well, they were deceived, but the violation is of the debriefing because they didn't debrief. Two that I put was confidentiality because they published the uncensored data, and then minimizing harm because if you notice before, they were being prescribed GLP-1 medications, and in this study, it didn't specify if they were like they needed the GLP-1 medications. Like taking the typical person taking Ozempic is harmful. Okay, can students say that GLP-1 medications cause mental health benefits? And at least give me like one piece of evidence. No, not statistically significant. Correlation does not equal causation. Probably wouldn't be a good answer to put, even though it's correct in this context, but you would have to explain that the effect size is the correlation and then this is the statistical significance. Um, I was in the experiment and it, oh, Anyways, no. So the easiest, this is probably the easiest one, but you're going to look at statistical significance first. Okay, so what are some possible limitations of this study? Only Asian women. So sample is definitely one of them. Generalizability, age. Yeah, so here is all of my possible answers. Um, this is also a good time to talk about weird, which is, what is it? White, educated, or what? White, educated, something Democrats. Definitely not literate. Guys, I'm sorry I had to Google this. Weird, um, psych. Okay. Weird is Western, educated, industrialized, rich, and democratic, which is basically saying that most of our psychological studies are actually conducted in the Western hemisphere of the world, which means that weird is an acronym to show what kind of people we're getting into our samples. So most of our psychological testing is all done on the acronym WEIRD. Um, Obviously, the country difference generalizability. Also, something important is we talked about effect size earlier, and in experiments, it basically shows the difference between the two conditions. And so there's a large difference, but it's not statistically significant, which means that it probably implies we have a strong confounding factor or multiple. Also, if you care, there was like a real research on this, but obviously not that for Q. Okay, let's get started. Unit 1. Okay, we're going to start with the age-old debate of nature versus nurture. The question we've always been asking ourselves about nature versus nurture is which impacts us more in whatever specific situation. To this day, we're still trying to study which plays a larger role in different processes, but here are some ways we've tried to answer that question. Am I literally? Oh, okay, I think I'm stupid. Oops. Anyways, nature is basically all your genetic characteristics, and then nurture are the external factors that impact you. Um, biological, sociocultural. Biological, psychological perspective. Okay. For nature, we can say biological and evolutionary. For nurture, we can say psychosocial and behavioral. The reason we're going to put behavioral in there is because social learning is a big part of behavioral. So, okay. Um, Firstly, the evolutionary perspective defends the nature or hereditary side of this argument. The evolutionary perspective is all about natural selection. Natural selection is Darwin's theory that basically says those with beneficial traits will be more likely to survive and reproduce to pass those traits on to their offspring. So, for the sake of this class, um, you need to know that natural selection and evolution is all about survival and reproduction. Isn't heritability of behavior above 50%? What do you mean? Um, Maybe. I know it's not in this unit, so Didn't study it today. Anyways, um, one way people have tried to unethically and unsuccessfully apply the evolutionary perspective is through eugenics. So, I need to remind you that nature versus nurture is an ongoing debate because we cannot fully separate them from each other. Eugenics is the disproven and unethical belief that we can improve humanity by encouraging the reproduction of those with desirable traits and discouraging the reproduction of those with um, undesirable, quote unquote, undesirable traits. A really infamous use of eugenics is the Holocaust. So, how are we able to separate genetic factors and environmental factors when we conduct research? Some ways we do this are through twin studies, family studies, and adoption studies. For example, identical twins share 100% of the same genes, and we can compare them to fraternal twins who only share 50% of their genes. In adoption studies, if the child is more similar to their adoptive parents, it may imply that environmental factors are a stronger influence on their behavior and vice versa. However, none of these factors can be controlled for the entirety of these people's lives. So even though we can say there may be an association, we can't determine a cause. Okay. By the way, when I use indents like this and this, that just means that it's a subsection of the thing that came before it. Your nervous system is divided into two main parts. Your central nervous system contains your brain and your spinal cord, while your peripheral nervous system contains all your other nerves that connect your CNS to the rest of your body. The two parts of the PNS or peripheral nervous system are the somatic nervous system and the autonomic nervous system. The somatic nervous system controls voluntary movements, while your autonomic nervous system controls involuntary movements such as your heartbeat and breathing. The autonomic nervous system is further divided into the parasympathetic nervous system and the sympathetic nervous system. The parasympathetic nervous system controls your rest and digest, while your sympathetic nervous system controls your fight or flight. For example, when you get really scared by something, your sympathetic nervous system is what's making your heart rate go up. Afterwards, your parasympathetic nervous system is what brings your heart rate back down to its resting level. Para, rest, digest. Whatever helps you learn. Oh, your nervous system contains two main types of cells. One of those is neurons, which are basically the cells that carry messages throughout the nervous system. They do this by sending chemical or electrical messages between each other until they get to their desired destination. The other main type of cell is glial cells, which work to support the nerve. This can be done by providing structure, insulation, communication, and voice transfer. In most areas of the brain, glial cells are neurons. Okay, parts of the neurons you need to know. Starting off, the cell body or soma contains the nucleus, which contains the DNA as well as energy for the rest of the neuron. The dendrites are branch structures that receive information from other neurons, while the axon is a long structure that carries the action potential or electrical impulse down to the axon terminal. The axon terminal is the part of the neuron that actually releases the signal to other cells, typically as neurochemicals like neurotransmitters. Um, there are two parts of the neuron you need to know that speed up the action potential: the myelin sheath and the nodes of Ranvier. The myelin sheath is this fatty layer that surrounds the axon. Kind of like, I feel like the most common acronym for this is the rubber insulation around a wire. The website where info is on 5Hive. Um, I made these slides like last week, but I'm gonna publish them after. I'll make sure onto lecture schedule. But I did make them by myself. Okay. Um, the most common acronym for the myelin sheath is kind of the rubber insulation that you see around a wire that helps the electricity conduct faster. The nodes of Ranvier are these little gaps that you see here between the myelin sheath, and that's basically where the action potential skips along. The synapse is a very small physical gap between neurons where chemicals are released and received by the other neuron. I have two side notes from this slide. You don't have to know this, but the oligodendrocyte is a type of glial cell, and that's the glial cell that builds the myelin sheath. So that's why you see that it's attached to the myelin sheath. Secondly, you've probably seen the axon terminals function defined as what transmits signals to other neurons. The reason I worded it as cells instead of neurons is because of something we'll talk about a little bit more later, which is called the neuromuscular junction. Some neurons don't transmit information to other neurons, but instead to muscle fibers. Okay, what is the reflex arc? The reflex arc is a pathway of neurons that rapidly stimulates a reflex action. The key to reflexes is that they're quick and they're involuntary. So if you see that anything in like a question that says immediate action, you can infer that it probably is a reflex. Because we need the reflex to be so quick, it bypasses the brain and happens in the spinal cord instead. Obviously, the brain is still involved afterwards and it gets the message that it happened, but it's not what controls the reflex arc. There are three different kinds of neurons in the spinal cord, or partially in the spinal cord, like you can see here, they're like partially in, partially out. The sensory neurons are neurons that actually detect the stimulus. In this example, the sensory neuron is carrying the information that the skin has been pricked and is in pain. The interneuron is the neuron between the sensory and motor neuron and interprets the sensory information to tell to the motor neuron. So here the interneuron is saying that because the skin is in pain, we need to get it away from the pin. The motor neuron then sends a command to the muscle fiber, telling it to pull the skin away from the pin. The neuromuscular junction is just a special type of synapse between a motor neuron and a muscle fiber. So right here. Sensory neuron, interneurons, motor neuron. Yeah, good. Okay. Neural transmission, which is basically how you fire your action potential. An action potential is the electrical charge that travels down the axon to send a message. The all or none principle basically tells us that an action potential is either firing or at rest. There's no in between. The threshold is the minimum amount of energy needed to trigger an action potential. Here we can see that these failed initiations are here because they didn't meet the threshold. Depolarization is when the electrical charge in a neuron shifts enough so that an action potential can be fired. After that, the charge dips below the resting state where the neuron cannot be stimulated for a certain amount of time. This is known as the refractory period. Two disorders that you need to know that are a result of neurotransmission being disrupted are multiple sclerosis and myasthenia gravis. Multiple sclerosis is when the myelin sheath around your neurons begins to deteriorate. Myasthenia gravis is when the communication in your neuromuscular junction basically stops working or becomes weaker. Okay, these are the only 8 neurotransmitters that you need to know for your AP exam. I can promise you. Okay, first we need to talk about the difference between excitatory and inhibitory neurotransmitters. Excitatory neurotransmitters increase the chance of a neuron firing an action potential, while inhibitory neurons decrease the chance of a neuron firing an action potential. The main excitatory neurotransmitter is glutamate, and the main inhibitory neurotransmitter is GABA. Neurochemicals are super complex, so you don't have to know if the other neurotransmitters are inhibitory or excitatory. Depending on the situation, they can work as either. Afferent and efferent, I'll go back. Yeah, afferent approaches, efferent exits. That's a really common, I don't remember what to call it. I just can't think. Also, afferent neurons are called sensory neurons and efferent are called motor neurons. Okay. Just so I'm not taking too long reading definitions, I'm just gonna let you guys screenshot that. But are there any specific neurotransmitters you want me to go over that are on this slide? GABA? Okay, GABA is our main inhibitory neuron, like we said, so that's the only neuron. The only neurotransmitter that you have to know is inhibitory. It basically calms activity, so it's important in things like sleep and rest. And when you release GABA from one of your neurons, it basically makes the next neuron less likely to fire. Norepinephrine. Adrenaline's different. Adrenaline is a hormone, and that is epinephrine. You do need to know the difference. It's okay. Norepinephrine is released with a lot of your fight or flight responses. So when you're scared of something, you typically release norepinephrine. Noradrenaline, I don't know if that's in the scope. Isn't noradrenaline a different kind of hormone? Not in the scope of this class. Um, Norepinephrine. Oh, well, I don't think the AP test calls it that, so you're probably going to see norepinephrine. They are both neurotransmitters and hormones, but for the sake of this class, norepinephrine is a neurotransmitter. Epinephrine or adrenaline is a hormone. Um, in reality, the difference comes from where they're released, but I won't be talking about that. Moving on. Okay, these are the only 5 hormones you need to know for the AP exam. Adrenaline, which is also known as adrenaline, which is also known as epinephrine, stimulates your fight or flight response. Leptin is a hormone that decreases your appetite and therefore is able to regulate your energy and appetite. Ghrelin is a hormone that stimulates hunger. Melatonin is involved in your sleep wake cycles. And oxytocin, you might have heard be called the love hormone. It's involved in bonding and social attachment. Oxytocin is super important during childbirth in order to make the mother strongly bond with their child. Yeah, leptin suppresses hunger or appetite, so it basically tells you you're full. Um, we are not tested on the endocrine system at all. It was taken out of the test from the old test, and you can Google it if you don't believe me. Okay, between neurotransmitters and hormones, which one travels faster? Neurotransmitters. Neurotransmitters. No one's team hormone. Okay, we're all team neurotransmitters. Yes. The example, not the example. The answer is neurotransmitters because hormones have to travel all the way through your bloodstream. Neurotransmitters just go through the synapse, so it's really short, and they're also bioelectricity, so already faster. Yeah. Okay. Let's talk about psychoactive drugs. Agonists are drugs that mimic or enhance a neurotransmitter's activity. For example, opioids like fentanyl and heroin are able to mimic endorphins and increase pain suppression. That's why we use them in anesthesia and in medical cases where the patient is going through a lot of physical pain. Antagonists are drugs that block the neurotransmitter from bonding. So it might latch onto the receptor for a period of time to stop the neurotransmitter from getting to the receptor. You're not going to be tested on this, but I think this is so incredibly important for like everyone to know. Um, One antagonist you should know is Narcan, which is an antagonist that basically knocks opioid chemicals out of the receptors and blocks them from reattaching. If someone is having an opioid overdose, you can save their life by giving them Narcan. So After you turn 18, you should look into getting licensed for Narcan. Reuptake inhibitors block reabsorption of neurotransmitters back into the sending neuron, which basically traps them in the synapse so that they have a higher chance to bind to a receptor, which increases the effect of the neurotransmitter. What are SSRIs? Serotonin reuptake inhibitors. What's the other S? Okay, well, selective serotonin. Okay, well, not in my slide, is it? Anyways, we use SSRIs to treat a variety of disorders because a lot of disorders have something to do with serotonin. So the SSRI will like block this channel and then it'll prevent the neurotransmitters from going back in. So it'll just stay in here so that they'll have a higher chance of attaching to a receptor. Okay, well, next slide. Okay, stimulants. These increase neuroactivity and they're drugs like caffeine, nicotine, methamphetamine, cocaine, and Adderall. Also, there is so much stimulant abuse. Reuptake. Reuptake is the process of a neurotransmitter coming, binding, and then it's the process of it going back into the sending neuron. Yay. Okay. Stimulants are drugs that increase neuroactivity, and this includes drugs like caffeine, nicotine, methamphetamine, cocaine, and Adderall. There is actually a lot of stimulant abuse in schools, like not just caffeine. Um, like there's a lot of high achieving students that like to use Adderall recreationally. So please, please, please don't do that. I went over agonists and antagonists, so you probably weren't listening. But okay. Um, you can screenshot this. Okay. So, but marijuana is a depressant. I'm fairly certain. Fairly. Oh, marijuana can act as a depressant. It can also act as a stimulant or hallucinogen. That's true for a lot of drugs, that they're able to act as multiple drug classes. Oh, back to why you shouldn't abuse Adderall or stimulants recreationally. You, when you abuse drugs long term, you can kill off your neurons and literally shrink the gray matter of your brain. You will shrink your brain. Most of the neurons you have today, you were born with, and you cannot regenerate them. Okay. Depressants are drugs that decrease neural activity, such as alcohol and a lot of sleep medications. Hallucinogens are drugs that can distort a person's perception and cognitive processes. Hallucinogens can be found in a variety of plants. So you've probably heard people say like shrooms, but you can also find them in just like cactus, a lot of cactus. A ton of plants have evolved hallucinogenic chemicals to ward off their predators, and in a lot of traditional native cultures, hallucinogens are used in ceremonies to make people feel closer to their ancestors or gods. Opioids are a class of drugs that relieve pain by mimicking endorphins and inducing euphoria by increasing dopamine activity. So they do both. Tolerance is when someone needs more of a substance to get the same effect than they used to. I need you to understand that tolerance is just a physical phenomenon. It can be a result of addiction, but not always. For example, if someone drinks socially every week consistently for a long period of time, they might begin to get drunk off of 2 glasses of wine instead of one. That's an example of them building tolerance, but it's not impacting other parts of their lives negatively, so it might not be an addiction. Addiction is characterized by a person actively seeking out a substance despite knowing the negative consequences. Withdrawal is a symptom of addiction where someone experiences discomforting physical and physiological effects without the substance in their system. So, some people experiencing withdrawal might get headaches, or they might get nauseous, or they'll have severe cravings. Okay, guys, I hate myself. You know what? I'll just read the question from what I remember. Why is the answer up? Okay, the answer is up. I'm gonna put it in chat because I like this question. I don't want you guys to, um, whatever. Okay, that's the question you guys have to answer. I accidentally put the answer on the side. Sorry. Who is paying? You guys are telling me the reflex arc is part of the nervous, is like a main part of the nervous system. Peripheral. Somatic. Your peripheral nervous system. I tricked you guys. Okay, I also tricked myself because I need to fix my screen. It's actually your CNS. Because, like we talked about earlier, your spinal cord controls your reflex arc, which is you jumping away and pulling your hand back, and it also controls the conscious action of you cursing her out. So make sure that you remember the reflex arc is in the spinal cord, and the spinal cord is part of your CNS. Obviously, you guys aren't wrong in saying that like the PNS is involved because obviously it's the thing that like is sensing all the stuff. But is it what's causing it? No. So just be careful and take questions slower sometimes, maybe. Okay, the brain. Okay, the cerebrum is the largest part of your brain and it includes your four major lobes, which we'll talk about on the next slide. But first, we need to understand that the cerebrum is split into two hemispheres. Contralateralization tells us that each hemisphere controls the opposite side of the body. The optic nerve is a super good example of this. You can literally see that the right visual field is processed in the left side of the brain and vice versa. So, like this. Okay. You also have a ton of nerves running between your cerebral hemispheres to communicate information. The largest bundle of those nerves is the corpus callosum, but you could probably get away with saying that it's connected by the corpus callosum. It's like this very dense um, bundle of nerve fibers. In patients with severe and life-threatening epilepsy, we might perform a surgery where we cut the corpus callosum. The result of this surgery is split brain syndrome. Lateralization describes how some functions are specialized in one cerebral hemisphere. One example of this is language, which is dominantly in the left cerebral hemisphere, typically. Both Broca's area and Wernicke's area are located on the left side of the brain. Broca's area is in the left frontal lobe and is what helps you produce speech. Wernicke's area is in the left temporal lobe and helps you with language comprehension. Aphasia is an impairment of language typically caused by damage to either Broca's area or Wernicke's area. Okay, here are the four cerebral lobes, and if you are to remember any anatomy diagram, you need to remember this one because last year one of the MCQs was quite literally this picture, and I don't remember if it was the parietal lobe or the temporal lobe, but that was the MCQ, and it asked you what lobe is this color. So make sure you remember this. Also, you should remember that a lot of brain structures are generalizations of functions. If you've ever seen a human or animal brain, you'll know that most of the brain kind of just looks like the same pink stuff. We determine what a lobe is by the indents of your brain, but you don't have to know that. But I do have to let you know that even though we say functions are localized to specific regions of the brain, that's not exclusive. Also, I said lobes every time here as in plural. You have two of each lobe because you have one in each of your two cerebral hemispheres. Your frontal lobe controls a lot of your higher level function. It contains your prefrontal cortex, which is involved in things like planning and decision making, and your motor cortex, which is involved um, with most of your voluntary movement. Okay, guys, my internet cut out, so I didn't get any of your chats. What's the pink cerebellum doing labeled? Because it'll be labeled on a different diagram. I'm not allowed to talk about past MCQ questions? Okay, too late. Guys, I take it back. I totally didn't see this question last year. Sorry. Okay. Your parietal lobes control your association areas, which are involved in higher level functions like learning and speech, etc. Um, okay, that makes sense because I didn't see you guys. Okay, where did I lag out? Was it the beginning of this slide? Yeah, but what part? Okay, guys, go back to the. Okay, I'm just gonna restart this slide. Anyways, please remember this diagram is what I have to say. Also, I need you to understand that all of these say front lobes, plural, S as in plural, because you have two of each lobe because you have one in each of your two cerebral hemispheres. Your frontal lobes control a lot of your higher level functions. It contains your prefrontal cortex, which is involved in things like planning and decision making, and your motor cortex, which controls most of your voluntary movement. Your parietal lobes control your association areas, which are involved in higher level functions like learning and speech. I don't want you to get caught up in that though. Like, if they ask you what lobe carries out higher level functions, you should probably say the frontal lobe. And if they ask you what carries out speech, you should say, guys, I forgot it now. You should say Broca's area. Okay. The parietal lobe also controls your somatosensory cortex, which is the place where your touch sensitivity is processed. Your temporal lobes are involved in auditory and linguistic processing, while your occipital lobes are primarily involved in vision processing. Okay, let's talk limbic system. Um, Anatomically, your limbic system is basically tucked inside your cerebral lobes. Some parts of your limbic system are partially inside of the lobes, though, but that's not really in the scope of this class. Your thalamus, I don't know why it's not labeled, but it's this dark navy blue region. Um, your thalamus basically is this train conductor of information inside your brain. It takes all the incoming information from your peripheral nervous system and sends it to different parts of your cerebrum for processing. Your hypothalamus keeps you in homeostasis by controlling a lot of your ANS and your hormones. Your hypothalamus is able to control your hormones because it controls the pituitary gland. The pituitary gland is basically the master gland of your entire body. The pituitary gland releases the growth hormone and it controls the other glands in your body. The hippocampus is a brain structure mostly involved in memory. The amygdala is a brain structure that is heavily linked to emotions. The amygdala is also in charge of the fight or flight response. Yes, true. Also is involved in emotional memory. Not that you need to know that much. Anyways, something that stuck with me last year when I was studying for this was I saw a flashcard set and it said angry almond, which is kind of silly, but it did help me remember because the amygdala is basically shaped like two almonds. The brainstem controls a lot of your basic functions that keep you alive. It also is what connects the spinal cord to the brain, so it bridges a lot of information. The three major parts of your brainstem are the midbrain, pons, and the medulla. You probably won't be tested on the midbrain, but it's involved in all sorts of things like auditory processing, visual processing, your arousal, and your motor movements. Your pons are highly important for your sleep, and your medulla controls vital things like breathing and heart rate. The reticular activating system, RAS, sometimes, is a network of nerves inside your brainstem that control alertness and eye movement. I'm going to go back to this slide. This is the cerebellum, this entire purple part. This is just the front part. This is the same part. The cerebellum means little brain in Latin because it looks like a little brain, but it coordinates a lot of your muscle movement and is involved in your balance as well. Reticular formation. Isn't that the same thing as reticular activating system? Okay. Yeah, there's multiple names for everything, but this is the one I took off of College Board, the document used for teaching and for the entire course. So I use those. Neuroplasticity is a super fun and fascinating topic. It's all about the brain's ability to reorganize itself by making new pathways after it takes a hit or like suffers an injury. Earlier we talked about split brain, which is the result of surgically cutting through the corpus callosum. Because of neuroplasticity, people with split brain are still able to function well because their brain adapts. Even though neuroplasticity isn't present in your entire life, it's a lot stronger in childhood. This is why sometimes in severe epilepsy cases, we can cut out the entire half of a child's brain. We normally can't do this to an adult because their brains don't have the amount of plasticity that a child has. Okay, so there's a variety of different tools we can use to understand. One way we can do that is by doing an EEG or electroencephalogram, which is where we measure the electrical activity of the brain in waves. We use EEGs when studying things like sleep or seizures, but we can also use it for other stuff. Eyes or functional MRIs basically plot out blood oxygen levels in the brain to show where the brain is the most active. Additionally, we're able to use case studies of people who suffered very unique brain injuries to help us understand where functions are controlled in the brain. So you guys like Phineas Cage apparently a lot, but he's a really famous case study. He was, I think he's a railroad. But anyways, he was involved in a construction accident where a metal rod shot through his skull and his frontal lobe. Even though he survived, his friends and family said that after the accident, his personality had changed dramatically. Because of that, scientists thought there might be a possible link between the frontal lobe and personality. Just to remind you, we cannot use case studies to generalize or infer causation. The reason that we know today the frontal lobe controls aspects of personality is because of extensive experimental research. We can also use surgery. We can use surgery such as lesioning, which is cutting out parts of the brain, and we can use that to observe loss or shifts in function, and note that there could be a possible link between a brain region and a function. Okay, sleep. Let's start off by defining consciousness. Consciousness is the awareness of ourselves and the things going around us in our environment. Sleep is a type of altered consciousness, which you need to know is distinct from unconsciousness. In reality, there's actually a lot of debate on if sleep should be considered a state of consciousness, but College Board says it is, so you're also going to say that it is. Unconsciousness is things like a deep coma or anesthesia. Circadian rhythm is what regulates our sleep wake cycles. It's kind of like a clock that tells our body what to do in 24-hour cycles. For example, our circadian rhythm is what tells our body to release the chemicals to make us sleepy during our bedtime. Two ways we can disrupt our circadian rhythm are jet lag and shift work. Your circadian rhythm also depends on environmental factors like how much light you're getting. For example, if you travel across the world and it's daytime during your normal bedtime, you can easily disrupt your circadian rhythm. Shift work is when people who work late into the night, or like a lot of people in healthcare that work late night shifts, they disrupt their circadian rhythm by going against it in order to work. Um, REM isn't conscious, but I have not heard of that. I don't know. For this course, you need to know that all stages of sleep are considered altered consciousness. So REM is would be a state of altered consciousness. Stages. We typically go through if you have a sleep normal altered consciousness. So consciousness um consciousness is like just a version of you said conscientiousness. Okay, we're talking about the big five. So did you mean consciousness? Um it's just when you're not, it's like the bridge between consciousness and unconsciousness, if that makes sense. Another, I think a better example of altered consciousness is drugs. So people um, that have taken hallucinogens are in an altered state of consciousness. Subconscious. No, you don't really need to know about Freud for this course. You also don't know, you don't need to know about his theory about dreaming either. But Freud is very interesting, so yeah. Okay, if you are a healthy person, um, then I probably misspoke and it's just not in this unit. So, yeah, I'll probably talk about consciousness later. Okay, we typically go through 4 to 5 cycles of sleep, and each of those sleep cycles lasts 90 to 110 minutes. We use EEG recordings to record the brainwaves and to identify stages of sleep. Firstly, I don't want you to misinterpret this graph. Having a higher spike like this or a wave doesn't mean that there's higher energy. You should be looking at how many waves there are in the same amount of time. So we see awakeness has the most waves in a condensed amount of time. Let's go over the stages. Non-REM is your initial transition from wakefulness to sleep. This is also the stage where you might have hypnagogic. Guys, I practiced this earlier. Hypnagogic sensations, which is the feeling of floating or falling or small muscle jerks. Hypnagogic sensations are actually super common. Like, an estimated 70% of people experience them, but we often just don't remember. Your EEG is going to show alpha waves during this stage. Non-REM 2 is going to show theta waves on the EEG. The sleep spindles are the short, rapid bursts of activity. So it's like right here. Oh, guys. It's like right here where you have a lot of waves in a short amount of time. And then your K complexes are this higher amplitude, slow frequency that kind of looks like stage 3. Non-REM 3 is your deep sleep and shows up as delta waves. You need non-REM 3 sleep to recover from physical and mental activity. It is super, super difficult to wake someone up from this stage. That's also why it's super difficult to wake up someone who's sleepwalking or someone who's having night terrors. REM sleep or rapid eye movement produces kind of similar waves to wake stage. Typically, this is when your body is actually the most relaxed because you just came out of deep sleep. Because of these opposites, we consider REM to be paradoxical. Um, Dreaming typically occurs in this stage, and because our bodies are basically in a state of paralysis from the deep sleep, it stops us from physically acting out our dreams. Typically, we wake up either in REM or non-REM 1, which is why sometimes we're able to remember our dreams. Doesn't REM dreams have theories for it? Yes. But I will cover the two you need to know, and Freud's you do not need to know. I think it's on the next slide, or it's on the next slide. It's coming very soon. Okay, REM rebound is a phenomenon that results from sleep deprivation of REM sleep. So this can either happen because you're not sleeping enough at all, like in general, or you keep waking up during REM, so you're not getting enough REM. REM rebound is when your brain will try to compensate for that by extending the time you're in REM. Oh, it's this one then. Okay, there are two main theories on why we dream. One is activation synthesis theory, which suggests that dreams are your brain trying to make sense of all the random neural activity in your brain during REM. So, for example, let's say your brain randomly fires action potentials and individual pathways that are related to books, the sea, and a sinking ship. Now it'll try to make a coherent story out of it. So maybe you're sinking on a ship in a sea of books or something. Memory consolidation theory suggests that dreaming is purposeful and is basically your brain's way to reorganize all of your experiences throughout the day to file into your memory, kind of like a filing cabinet. These are kind of like subsects of memory consolidation theory, but they basically are theories that explain why we sleep. So, memory consolidation happens in the hippocampus during deep sleep or non-REM 3, where memories are stowed away for us to remember later. Dream theory isn't tested, right? Yes. Memory restoration is the idea that sleep basically helps us restore energy and reset our mind so that it's ready for the new day. Memory consolidation and memory restoration show us why it's important to get sleep in order to function and learn. Okay, let's take a quick look at sleep disorders. Insomnia is characterized by difficulty falling or staying asleep. Insomnia can be triggered by stress or just bad sleep hygiene, so it can be long term, but it can also be short term. Narcolepsy is a biological and chronic condition where someone has uncontrollable sleep attacks during the daytime. Most people with narcolepsy will experience cataplexy, which is a sudden onset of muscle paralysis while they're awake. REM sleep behavior disorder is when you don't have the same muscle paralysis most people do during REM, causing people to physically act out their dreams. Sleep apnea is repeated stops in breathing during sleep. This can happen because this can happen because your brain is either failing to tell you to breathe or because your airway is physically blocking you from getting oxygen. So, people with obesity are at a super high risk of developing sleep apnea compared to um, not over not obese people. I don't know how to word that. Okay, people with sleep apnea typically have low quality sleep because those stops in breathing can wake them up periodically. I'm not gonna lie to you, I don't know how to pronounce that. You do need to know both um, terms of sleepwalking and somnambulism. Guys, hopefully you know how to pronounce it. Not that you have to. But, anyways, it's when people walk around or do all sorts of activities during deep sleep. Sleepwalking is most common in children and is something a lot of people grow out of. Um, oh, can someone tell me why sleepwalkers don't remember sleepwalking? Because their brain is inactive, because N3, non-REM 3. Yeah, pretty much. So deep sleep is characterized by really low brain activity. So they're not gonna remember it probably. Also, I googled this earlier today because I was curious. It's not actually like harmful. The act of waking up someone who's sleepwalking is harmful because like they can scare themselves and maybe they'll accidentally like hit something. But like the act of waking them up won't cause them any harm. Okay. Um, First, we need to define sensation. So, sensation is the process of detecting stimuli and converting them into neurochemicals which our brain is able to perceive. The absolute threshold is the minimum intensity needed to be able to detect a stimulus 50% of the time. The just noticeable difference is the minimum difference needed between two stimuli to detect that they are different 50% of the time. Weaver's law says that mathematically, the the just noticeable difference is based on proportion of the intensity of the two stimuli instead of a fixed difference. Um, why do I feel like I'm about to fall out of my bed and I scare myself even though I'm. Wait, I need to reread that. Why do I feel like I'm about to fall out of my bed and I scare myself even though I'm in the middle of my bed? That is a hypnagogic sensation, which is your, in non REM 1. Yeah, okay, back to sensation. So, oh, an example of Weber's law. By the way, I very much made this up. The numbers are very false. Um, so, Weber's law would say that if I have one light bulb that was emitting more light than another, the light bulbs would need to have a difference of 20% of their energy rather than saying one needed to be 20 lumens weaker than the other. Sensory adaptation is when we become less sensitive to a repeated stimulus. For example, if you're sitting somewhere quiet and people begin to talk around you loudly, even though it might bother you a lot at first, you'll become less sensitive to it over time. Synesthesia is a lifelong phenomenon where someone's sensations sort of get kind of all jumbled together. One kind of common type of synesthesia is when people experience sounds along with colors. Can you explain Weber's law again? Yeah. So Weber's law is about the just noticeable difference, and it says that mathematically, the just noticeable difference is based on the proportion of intensity between the two stimuli instead of a fixed difference. So my example was that um, Weber's law would say that the light bulb would need to have a difference of 20% energy rather than saying, oh, it needs like this many volts more, this many lumens more. Okay, the eye, I'm not going to go over all the parts of the eye, but by the end of the slide, if you still have questions about parts of the eye, just let me know. Your blind spot is the part of your eye where the optic nerve is, so right here. We don't sense our blind spot because our eyes combine the images from both our eyes to fill in the gaps. If you close your eye, you've probably done it before, but like if you close your eye and you hold your thumb out, you can find your blind spot. Accommodation is the process where the ciliary muscles, so right here, this thing kind of attached to the lens, these muscles will push and pull to bend your lens in order to focus on far or near objects. If your accommodation is not working properly or your eye is just unfortunately shaped in a way that will give you nearsightedness or farsightedness, you can end up with one of these issues. So you don't have to know this, but like one example of that is like an astigmatism. A lot of people have um, astigmatisms where their eyes are like football shaped or something like that. That can give you nearsightedness or farsightedness. Yeah, astigmatisms are super common. Okay, the retina is this back layer of your eye, um, and that's where light is focused and transduced into electricity for your brain to interpret. The retina contains your rods, which are light sensitive receptors, and your cones, which are color and detail sensitive receptors. The fovea, which is right here, is this little tiny point in the retina where there are no rods at all, and that's where most of your cones are, which is why your vision is the sharpest there. Hopefully, if you remember that, that will also help you remember that cones don't just do color, but they also do detail. Does anyone need to know anything about any other parts? You probably won't be tested on but myopia, hyperopia. Myopia is nearsightedness. Hyperopia is farsightedness. Lens, the iris, maybe the pupil. What's the other? The pupil. Well, I'm asking them what they're asking about. Do you mean the pupil or something? Pupil? Okay, the pupil isn't pictured here, but the pupil is this. Do you see this gap right here? The pupil is basically just a hole. What happens with light? So let's have a ray of light coming here. It'll first refract through the cornea, and then it'll pass through the pupil, and then it'll hit the lens. And the lens, which we talked about earlier, can do accommodation, which means that it'll bend or retract itself into different shapes to be able to focus on close versus far objects, kind of like a camera lens will. Um, you can, the reason your pupil gets bigger is because um, your ciliary muscles can control how big this gap gets or how small it is. And we strain our eyes looking at things that are far away or things in dark spaces because our ciliary muscles are um, pulling the pupil to get bigger. Yes, you need to know that just carries it to like your occipital lobe and places that what's it called? That do visual processing. Also, your blind spot is there. You need to know that. Okay, hopefully that's good. Okay, let's talk about color vision. We have two main theories of color vision, which is one trichromatic theory and then opponent process theory. We said we have cones. We have blue, green, and red cones. If you haven't taken um, a science class before or learned this in a science class, visible light colors are distinct from each other because they have distinct wavelengths. So you do not have to know, you don't have to know like the specific wavelength number, but you do have to know between these three which has the highest and which has the lowest wavelength. So blue light has the shortest wavelength, green light medium wavelength, and then red has the longest wavelength. Yeah, blue has the shortest out of these three. Out of these three, which are the only three you need to know. Okay, trichromatic theory basically proposes that we see colors by mixing together the signals from these three cones. In the retina, we also have ganglion cells, which are the neurons that take information in from our cones. The opponent process theory suggests that these ganglion cells organize our color vision by pairs of opposing colors. Um, if you don't know this, you should look at a color wheel, but you have a red-green pair, you have a blue-yellow pair, and then you have a black and white pair. Blue-orange, it's blue-green. I'm fairly certain, maybe. Yes, you do need to know your pairs. There was a ping. Can someone tell him there was a ping? Okay. Opponent process theory says that when one color in the pair is activated, the other color is inhibited. So, one example is if we stare at this red car for too long, we can tire out our neurons that activate the red complex. So, when you close your eyes after and it brings you to black, which is a neutral colored background, you might see an after image of a green car. Blue, yellow, green, red, green. Blue, yellow. Um, I did I say blue? Did I say blue green on accident or something? I thought I said blue yellow. You have a red green pair, a blue yellow pair, and a black white pair. So if you look at a color wheel, you're going to see that red and green are on opposite sides, and then blue and yellow are on opposite sides. Also, you can tell from this picture red, green, blue, yellow. And then I feel like black, white is kind of intuitive. But yeah, that's your pairs that you need to know of ganglion cells. Color blindness is a result of damage to either your cones or to your ganglion cells. Dichromatic color blindness is when you have two working types of cones. Monochromatic color blindness is when you only have one working cone or you have no working cones at all. So this is really rare, but that's people who like see in like plain black and white pretty much. The most common type of color blindness is red-green color blindness, which is dichromatic and caused by a dysfunctional red cone. You also need to remember that cones do detail. I forgot how to pronounce this, even though I practiced it earlier. Hopefully, I will again. The two visual disorders you need to know. You guys can Google it because I forgot that. I actually struggle with words so much. Anyways, it's that, and that's also called face blindness, where someone is unable to recognize faces. Some people describe this condition as seeing people's facial features as puzzle pieces, but not being able to piece it together. I would call it face blindness, but I'm fairly certain it will show up as this and not face blindness. Although if you the thing with multiple names is you can totally use the other name that might be easier for you to remember on an FRQ, but you're probably gonna see this on the MCQ. In this picture, someone with facial blindness would not be able to recognize this as different people because all they see are the identical individual features. So they have issues with like sensing space and whatnot between the features. Blind sight is when someone is blind in one part of their visual field, but they're still able to respond to visual stimuli. This typically happens when someone's occipital lobe is damaged, but the other parts of their brain that process vision are still working, so they can still respond to the stimuli as if they could see it. Okay, let's talk about someone in here. Here is a diagram of the ear. The important things you need to know. You don't need to know the big bones. You don't need to know the eustachian tube. Not really. Yeah, I would say, and you also don't need to know pin. But the other stuff you should. Um, Basically, sound waves will travel through the loop into your ear through vibrate in your eardrum, and then they'll end up in the cochlea. The cochlea is this kind of seashell shape, and it's filled with fluid and covered in tons of tiny hairs. Those hairs are what convert sound waves into electricity, and then they send that electricity through the nerves into the brain. Pitch for sound wave frequency is about high or low sound waves. Loudness is the amplitude of the sound wave. Phase theory suggests that specific frequencies are heard in distinct regions of the cochlea. Volley theory suggests that neurons take turns firing in super quick succession to keep up with high frequencies. Frequency theory suggests that the pitch we hear is entirely dependent on how fast our neurons fire. Sound localization is just referring to our ability to tell where a sound is coming from. Well, I'm going to call it auditory nerve right now then. So, I don't know. You're probably not going to see it be called that on the AP exam. But you also probably won't be asked about that specifically. Okay, sound localization is referring to our ability to tell where sound is coming from. One way we do this is by processing which ear the sound is louder in. So if I hear something that's louder in my right ear, I can infer that the sound is closer to my right side. Hearing damage can be a result of just aging or damage to parts of the ear. Conduction deafness is when you have a problem with the outer or inner ear, so before ever, oops, outer or middle ear, not inner ear. Outer or middle ear, so before it even reaches the cochlea. Typically, this is like if something is jammed up your ear, or if there's like a ton of earwax in your ear, you can get conduction deafness. So typically, we can fix it. Sensory neurodeafness is characterized by having an issue with the hairs on the cochlea or the nerves and is usually permanent hearing loss. You can get this by damaging your ears by hearing a really, really loud and damaging sound, or you can also get it from aging because as you age, the hairs off your cochlea kind of die and fall off. So, sensory neurodeafness as you get older is normal. Okay, let's talk about your chemical senses, which are olfaction and gustation. Olfaction is your sense of smell and is the only sense that does not go through the thalamus first. If you don't know what the thalamus is, it's like the train conductor that takes all the senses from your peripheral nervous system and then tells them in what parts of the cerebrum to go to. You also need to know that pheromones are chemicals released by humans that send chemical messages through your olfactory system. Gustation is your sense of taste, and there are 6 flavors that you need to know: sweet, sour, salty, bitter, umami, oleogustus, I think. Hopefully you know the first 4, but umami is kind of the savory taste, and then oleogustus is the taste of things like fats and oils. I typically hear it be interpreted as savory, but there's like different interpretations. On our tongue, we have a lot of taste receptors, and depending on the amount of taste receptors on someone's tongue, we can classify them as non-tasters, medium tasters, or super tasters. So someone who is a non-taster is going to have pretty low amounts of taste receptors, and someone who has high amounts of taste receptors are going to be super tasters, where they're super sensitive to taste. Also, taste is a combination of olfaction and gustation. So this is why when you're sick and you have kind of like a stuffy nose, you might feel like your food is less flavorful. Okay, touch is processed by various structures in your skin and in your brain. For example, when you feel something that's hot, there's a combination of activated warm and cold receptors on your skin. Gate control theory proposes that there's basically this like theoretical gate in your spinal cord that opens and closes to dictate how much pain your brain can perceive. Pain receptors will open the gate, but touch receptors and the brain can close it. For example, let's say you get a paper cut. Um, if you're able to distract yourself, that's an example of the brain closing the gate. If you put pressure on top of the paper cut, typically that can relieve pain, and that's the touch receptors closing the gate. Yeah, you can also rub, like pressure, stuff like that, all in your touch receptors. Okay, phantom is XX. Phantom limb sensation is when people who have had limbs amputated still feel sensations or pain from where the limb used to be. This proves to us that pain is not entirely physical, it's also processed in the brain. Your vestibular sense is what controls your balance and is mostly detected by parts of your brain. And the semicircular canals in your ear, which, if we go back and you don't remember, is this. So you do need to know that. Kinesthesis is basically just describing our ability to know where our limbs are and what position our body is in without looking at it. Is that why I can't feel my legs after. Wait, is that why I can't feel my legs and I can't walk after I get out of the bathroom? No, I don't think so. What were you referring to? Yeah, I don't think any of these are gonna do that for you. Anyways, that's it, guys. So, I have a survey for you guys to take. I'm gonna put it in chat. It's super important for me and for Five Hive. So, yeah, super short. Please take it. What did you get on the exam of 5? Um, yeah. So I'm gonna do